so cool. All right. All right. Housekeeping. Lab today, we've got some presentations to hear. Got yours. Crypto. Evasion.
Yeah.
Classic question at the blood donor's office. Have you ever tested positive for babesiosis? They added that several years back. We also have a presentation on Saculina. I think that's today. What's that?
Which one's that?
It is a it is a it is, see here, oh, classic, rhizocephalin. It's a crustacean, I believe. It infects crabs, and you're going to see pretty impressive post-behavioral manipulation. So that should happen today. We'll have a quiz in lab, including some of our plasmodium, some of our AP complexants. So give you a warning that it's not multiple choice, it's fill in the blank. This will be a real test. What do I remember?
Looking over instructions again?
Yeah, and also identifying the parasite. What a parasite is. So it'll be a good test because we have this week of lab and next week of lab, and then we'll have our practical. I mean, semester's almost over. Can you believe that? Also posted quiz for introduction to the protozoans and the African trypanosomes. I set the due date at the end of Wednesday night, so you can kind of review it. You know, nice thing about the African trips is they have a pretty simple life cycle, and it's just the one life cycle. The difference that we have is the host, right? Trypanosoma brucei is in cattle. Brucei gambiense and rhodesiense are in humans. So make that easy. All right, we've got Leishmania is where we left off. We did our life cycle. If you remember, that life cycle is kind of much more in depth than we have had in other life cycles. Yeah, some of the life cycles are like, well, you have your acanthor burrows out, gets into the hemocoel, develops the acanthellum, and so forth. I mean, we've had some longer life cycles, but by far this is probably the most complex because you have replication that's going to take place in the vertebrate host. We know that, all right? But it's going to occur in either lymphoid cells or macrophages, cells of the reticular endothelial system, so the phagocytic cells. And then in the invertebrate, we have two places where replication is going to take place. It takes place, first place is in the mid or the hindgut. That's going to be species specific. And then it moves forward and it moves anteriorly up to the foregut, where it continues replicating a bit. And then you have this transition to the metacyclic form, but also this other form called these haptomonad parasite spheres that are circular in nature, and they attach to the lining of the stomadeal valve. But on the first case, that once we get to that spot, we're not necessarily infective, and actually transmission's pretty low if our insect never again takes a blood meal. So this is where, when that insect takes another blood meal, the blood and the plasma seem to trigger transition of that metacyclic stage back to a replicating promastogote stage. And when you have that replicating promastogotes, some of those will become infective trypanomastogotes. Some of them will transition to the haptomonad parasite spheres, and all of them will release this promastogote secretory gel that helps to form the plug in the fore and the hindgut, so basically in the area of that stomatidal valve. So when our insect then bites the host, it's going to have to first regurgitate that plug before it can start to take a blood meal. All right, so this is all aimed, all of this is, the formation of all of this is all aimed at getting these metacyclic trypanomastoids that are in the foregut into the next host. And the parasite has kind of found a way to plug up that anterior portion and cause that insect to regurgitate right into our host. Something that's pretty crazy. All right. Any questions on the life cycle? All right, immune evasion. So, the African trypanosomes, how did they evade the host immune system?
Different expression.
VATs, right? Determined by the VSGs. So you just change it, periodic antigen types, and those VSGs help to protect the conserved epitopes. All right, how did the American trypanosomes evade the host immune system?
Alter the response.
Yeah, immune modulation, right? IL-7, is that right? IL-7 responds to IL-2. Immune modulation, right? We're basically reducing the cell-mediated response, right? Because that's effective against our parasite. Leishmania has a different way. All right? So this parasite lives inside of the cells that should be clearing it, that should be killing it. What we've seen, and there's several things that this will do to kind of aid in its life cycle. But what we do see during its life cycle is it has a change in surface coats. It has a change in the thickness of the surface coat. So this is something that, electron microscopists, they were looking at your promastogotes, your trypanomastogote stages in the insect and the vertebrate. And they're noticing that surface coat is different thickness. So that has prompted them, other researchers then to look at what's going on. Why do we see this change? Now, how big of a change is it? Procyclic promastogotes, 7 nanometers. These aren't going into the host yet. The metacyclic promastogotes have more than 17 nanometer surface coat. All right? We've increased it, which tends to suggest then that maybe the surface coat is important in survival of this parasite. But most importantly, when we look at the amastogotes, that surface coat is almost absent. So the question is like, okay, hold on, what's going on here? What are we using that surface coat for? So the surface coat, did you guys get this? Because we're going to go into the molecules. To just kind of demonstrate what's differences. Big surface coat, almost no surface coat. So there's a couple surface molecules that are really important. All right, ready? All right, so in the promastogotes, we start to see an increase in two things, LPG and GIPL. So LPG is lipophosphogycans. They're going to coat the surface, and they're going to continually shed it. So it's going to be on the surface, they're going to lose it, and they'll just keep shedding it. And this gets increased as we transition to our metacyclic trypanomastogote form. This is part of that 17 nanometer, or more than 17 nanometer thickness. What is its role? Well, it's got two roles. Number one is it inhibits complement-driven lysis. So one of our immune responses that utilizes the complement system to come in and basically poke holes into the parasites, pokes holes in the invaders. This is going to inhibit that response. This is kind of important because they're not in the macrophages yet. They need to get to the macrophages. So this inhibition is kind of extending the time that they can survive until a macrophage picks them up. But they're not out of the woods, because once they get into that macrophage, or once the macrophage phagocytizes them through whatever process is used, they're still susceptible to the hydrolytic conditions that end up forming in the phagosome lysosomes. All right, so you get your phagosome, you have fusion with other endosomes, including lysosomes that reduces your pH, activates enzymes, and it kind of destroys and enzymatically breaks down whatever's in that phagosome. These promastigotes can get destroyed that way, and this is where LPGs actually inhibits that fusion. It buys them time until they can transition to the amastigote stage. Once they get to that amastigote stage, they're in the clear because the amastigote surface is basically immune to whatever conditions are in that phagosome lysosome. So the LPG, these lipophospholipase inhibits complement-driven lysis, allows them to get into the macrophages, and then once they're in the macrophages, it delays that fusion that would be very successful at eliminating the parasite. The second surface molecule is GIPL. This is glycosyl and acetyl phospholipid. GIPL. I probably will just use LPG and GIPLs. This is the most abundant surface molecule. It's more than 10 times as abundant. It works out this way because it's super small and it's super close to the surface. So you can have a lot of it. This is not shed. Right, so it's going to be retained. Now its actual role is unknown. But researchers think it does have some sort of protective role. Maybe it's there to capture, I don't know, maybe antibodies, maybe it's capturing other cytokines, inhibiting signaling pathways. It's on there. It's abundant. But we don't quite know what its actual role is. All right, so those are the promastigotes and the trypomastigotes. So as we're getting into our vertebrate host. What about the amastigotes? Their surface coat is virtually absent. Doesn't really need it because it is pretty resistant to the acidic and hydrolytic degradation that would occur in that phagosome lysosome. So we know that once we transition to the amastigotes, both LPG and GIPL are downregulated. I think this part is partially the reason why they think GIPL is important and is playing some sort of protective role. Now, the amastigotes aren't completely immune to everything. Reactive oxygen intermediates can destroy these things, so that's also part of our immune system. And we're going to hear about these again with Plasmodium. But these reactive oxygen intermediates, can actually damage the parasite. So we do know that these amastigotes will suppress the production of those intermediates, so that they can hopefully try to evade that type of response. So for Leishmania, it's all about kind of resisting the immune system, resisting processes until it gets that amastigote stage, and then it's pretty much in the clear in that phagosome lysosome, that phagol lysosome. Ready? All right. So Leishmania, as I said, we've got 3 main species. Life cycles are similar. We do have differences in what cells they're primarily infecting, and also where in the intermediate host they're going to replicate. Now, for the intermediate host, we're not really going to distinguish between the mid and the hindgut. All right, they're just going to, depending on the species, who cares for us? But in the vertebrate host, we do need to know what cells they're infecting because that can influence the pathology that we see. So first up is Leishmania tropica. This parasite's found in Western Central Africa. It's found throughout the Middle East and Asia Minor, as well as in India. Leishmania tropica is responsible for cutaneous leishmaniasis. It's a cutaneous form of leishmaniasis, which means our parasite is going to be infecting and replicating in the lymphoid cells of the skin. Lymphoid, I missed an H. Get that corrected. Lymphoid cells of the skin. Right, good. Ooh, the audio is a little fuzzy. See that? Sorry. All right. Hopefully this is a little better. Everyone Colin said the audio was a little fuzzy, so hopefully it's just a hiccup. But anyways, Leishmania tropica, cutaneous form of leishmaniasis. We're infecting the lymphoid cells of the skin, and when we damage them, we will actually cause the skin to open up and exhibit an open wound. All right. This cutaneous form of leishmaniasis has taken on several different names. It's been called oriental sore, right, if you think about Asia Minor and India, right, for that area, as well as Jericho's boil. Right, so cutaneous leishmaniasis has taken on two different names. Now, this will heal. The scar will, or the lesion, that sore will heal, but it'll leave a scar. And as it does heal, we do acquire some protective immunity. So if we get it again, our body oftentimes takes care of it before we develop the open sores. Are there ways to treat it? Yeah, there's different ways to try to treat this parasite. One way involves heat. Because the parasite's so close to the surface, they are near the edge of our sore. If you apply heat and degrade protein, get it up to like 140, 150, just below the point where we're burning the skin, you can kill off the parasite. At least that's been experimental treatment. How we can administer it and be cost effective in administering it, that's another story. All right, so cutaneous leishmaniasis, Leishmania tropica. Questions on this? Next up is Leishmania donovani. This is, we said the amastigotes have taken on the name of a Leishman-Donovan bodies. This is part of who found it, so you got their name, Leishmania donovani. This is found in India, China, and Bangladesh. Donovani causes visceral leishmaniasis. This has also taken on several different names, including Kala Azar and Dum-Dum fever. Can't help but think of Night at the Museum. Definitely, Dum-Dum fever is based on the location, the area where people thought you were getting it. All right, so the visceral form isn't going to form these skin lesions. The visceral form is going to be infecting your macrophages, cells of the RE system. All right, and what you're going to end up getting is infection of the spleen, of the mesenteries, of the liver, lymph nodes, intestine, all these different places where you have these phagocytic cells. The result is progressive wasting. You're going to see anemia because of the destruction of the cells that are happening when our amastigotes rupture them, as well as development of hepatosplenomegaly. It's the enlargement of the spleen and liver. And you get a massive enlargement. So individual here is they've kind of put up, how big the spleen and the liver will be. I mean, it's massive enlargements. We've seen this before in what other parasite? Splenomegaly. What's that?
The one that geophagy was involved with.
Okay. Why would we exhibit geophagy? What? The iron deficiency. All right, so what parasite could cause iron deficiency? Good review for the fun. What feeds in our blood? Okay. Also, schistosomiasis can cause this as well. Causes symptoms. So just because you see splenomegaly doesn't mean you have one or the other. Several things can produce it. All right, if left untreated, this parasite could ultimately cause death. Now, what's kind of interesting is that you can get treatment, but the treatment might be incomplete, or you may have, what I'll say is clearance. Your immune system appears to clear, and then later on you develop this thing called post-Kala-Azar dermal Leishmanoid. This is where the parasite's still visceral, but it's kind of moved closer to the surface, where it's forming these reddish, depigmented granules or nodules. So you can kind of see it here. They lose their pigments, and they can continue to get large. That leaves, basically leaves you with major disfigurement. This can occur if you got treatment, but the treatment maybe wasn't administered long enough. And then unfortunately, this can happen with any of our protozoans, where you get it treated, and the treatment option may say you need to treat for 14 days, but doctor only prescribes 7 days. All right, so this is Leishmania duodenalis. Any questions on this? All right, our last one is going to hit closer to home. Leishmania brasiliensis. This is South and Central American parasite. And this is South and Central America. This is one that if you go on that summer trip, tropical biology trip, oh yeah, you can be exposed to this. You can get it. All right. Now,
The students got it before?
here, maybe. Trying to remember, former student. I know he came back, he's had bot flies. And I'm trying to remember, I think he had it on his foot. Went to grad school with a kid who worked down in the Andes, Amazon. He came back with this. So what's important about brasiliensis is that it initially starts out as a cutaneous form of leishmaniasis. And it's typically right at the site of the bite, wherever you get bit. That's where you get that lesion forming. The parasites are in the lymphoid cells of the skin, causes the lesion to form, and then it takes kind of a long time for it to clear up without treatment. So it'll take 6 to 15 months. Once it clears up, then, you didn't really kill off the parasite. The parasite's still in the body. What then develops is something called mucocutaneous leishmaniasis. This is a thing called Espundia, it's called Uta. It's taken on several different names depending on the country in which they're identifying, or in some cases, which Central or South American Indian tribe, was reporting on it. So there's numerous names for it. But what has happened is the parasite's gone from the lymphoid cells of the skin to start attacking the cartilaginous tissue and the soft mucous tissue, usually in the nasal system and the buccal nasal system and in the mouth region. And this results in pretty severe destruction, as these images display. It's basically going to destroy tissue. So this can develop while the lesion is healing, or it could develop later on after the lesion has already been cleared and you think you're in the clear. So this is naturally, it'll recur, relapse. In some cases, it happens due to inadequate treatment. So the treatment clears up the lesion, but it still didn't kill off the entire parasite. So the parasite then comes back in this mucocutaneous form. That's what happened to the grad school when I was at Wake. He had the cutaneous form on his forearm. It was just that one lesion, and it got pretty big, silver dollar size. Right, we knew what it was. Right, the department knew what it was. But this is a tropical disease. Only the CDC could diagnose it and prescribe the medication. So you had to wait for that week to 2 weeks before it was confirmed. And then when his doctor treated it, they did not treat it for the proper number of days. So I think the treatment was supposed to be 2 weeks. They treated him for 10 days. And that was something my professor had said, hey, you need to contact your doctor. You're not getting treated long enough. I don't think they cared. The lesion cleared up, and literally, oh my goodness, two months later, it came back in the mucocutaneous form. His face swelled up. He lost part of his nostril from it. And at that point now, your treatment options are much more severe. He had to have a drip, an IV drip on his arm at all time. Caused some pretty significant side effects to him. But eventually he cleared up. His face swelling went down. But he's still missing part of his nostril. So, problem with Braziliensis is that it utilizes many, many reservoir hosts. Common ones are probably like sloths and anteaters down there. So, even if you go out and treat everyone, all humans, you still have this issue of Braziliensis in these other animals. Questions? All right. So this is going to wrap up our kinetoplasts. We still have flagellates, but we've wrapped up our kinetoplasts. So these are all the ones that had the kinetoplasts in the mitochondria, had your trypomastogotes, promastogotes, epimastogotes, and so forth. Note, hopefully you're seeing all these different names, disease names. Yep, we'll have some of those where I'll ask you, okay, what parasite causes Kala-Azar, and you'll need to know is Leishmania donovani. All right, no, symptoms, what we're seeing. All right, questions? Let's move on. So, we're on flagellates. Flagellated protozoans. All right, so we're going to do the others. And this will include two phyla, the Retortamonada and the Parabasalia. In simple terms, this is Giardia and Trichomonas. All right, so I'll introduce those. All right, Retortamonada. Features of this phylum is that they lack mitochondria, really don't have the need for them, and they lack dictyosomes, which is like, what the heck is dictyosomes? Again, it's our nice microbiological way of saying Golgi. I'm gonna slam those microbiologists. They always have to come up with their own terms for every single group. Because parasitologists never do that, right? All in different larval stages. Yeah, so they lack mitochondria and dictyosomes, which kind of has an implication. So if you think about it, if you lack mitochondria, then you're not taking part in oxidative phosphorylation, which means your ATP generation is going to be primarily through glycolysis or substrate level phosphorylation. And if you don't have Golgi, you're probably not going to be processing and secreting a lot of proteins and stuff. All right. So members of this group also possess recurrent flagella. that are found in a cytostomal groove. Now, the recurrent flagella are flagella that can extend backwards. Right, so I have this diagram here where we set up anterior is up at the top and the posterior is down at the bottom. These are recurrent flagella are those that protrude forward but then extend backwards. That's a recurrent flagellum. And a cytostomal groove is where the flagella typically extend. So instead of emerging right at the posterior surface where it comes out, it's actually emerging inside the groove, and the groove then forms kind of like a channel. We don't really see that in our own species, in our Giardia. And then they occupy anoxic environments. Helps kind of explain why we don't have mitochondria. All right. So this is a fairly large group. All right. The only one that we're going to talk about is Giardia. So this is in the class Diplomonada, in the order Diplomonadida. And features of this order is that we have two identical nuclei laying side by side. All right, so in our Giardia, we've got two nuclei. They're going to be side by side. Members of this group also have anywhere from 1 to 4 flagella per nucleus. So you could have as many as 8 flagella total. All right. Giardia is not the only parasite in this file, but it's the only one that we're going to talk about. All right. So for us, it's Giardia duodenalis or Giardia lamblia. Could be also Giardia intestinalis. All right. You've had multiple names. They've all been basically merged into this one, which I think the most common way you'll see it is Giardia duodenalis. Older ones might be Giardia lamblia. Right, this parasite was described by Antony von Leeuwenhoek in 1681. Where have we heard that guy before? Microscope, right? Microscope guy. Where do you think he identified these?
Oh, goodness.
His own feces. That's dedication. Dedication for you. Not some other person's or not some dog's feces. His own. Which is... yes, we'll leave it at that. All right. So as I said, several species have been synonymized. You're going to see them all through different parts of literature. So if you look in some of the older books, if you see Giardia intestinalis, don't think that's different than duodenalis. They're all basically the same. This parasite's cosmopolitan parasite of humans. Affects everyone globally, but it's most commonly found in warm environments. Most commonly found in warm environments could be due to our behavioral changes in the warm environments. We're more likely to spend time outside in aquatic habitats that would expose us to the cysts. But that's just speculation. All right, ready? So Giardia is the most common flagellate of the human intestinal tract. It's notable that we can detect it in about 81% of the raw surface water. It's abundant. It's prevalent out there. Now, we do see differences in prevalence in humans, and it varies by region, by geographic region. In some cases, it could be as high as almost 16%. Other cases, it may be down to 10-15%. Some of it may be due to our association with reservoir hosts, including, let's say, wild animals like beavers. It could also be how we clean the water, purify the water, if we even do. Higher densities of people are likely to have higher prevalences too. This was a trophozoite. You probably recall looking at that in the lab. Here's our cyst, double nucleate. All right. Ready? All right. Life cycle. I tell you, our life cycle for Giardia isn't terrible. The life cycle for Trichomonas is a whole lot easier. I won't even draw it. But for Giardia, I think it's probably good to draw it and get important stuff up there. So we are drawing Giardia. Make sure the video up here. Yep, Giardia duodenalis. Alright, so we have our trophozoite stage. It's going to be the dominant stage in our gut. Alright? And this is going to be found in the small intestine. This is where it's going to attach and replicate. It's going to replicate by binary fission. And it happens pretty quickly. It could happen so quickly that the vast majority of your surface, maybe the entire surface of the small intestine, gets covered by these guys. They're going to replicate. Eventually, some of them will be moving into the colon. And that movement's probably not, well, we know it's not a conscious decision. They're not going to be saying, well, maybe it's time to move. They don't do that. I'm also not saying that it's like programmed where they are designed to move. This movement in the colon is probably a consequence of some of the pathology that they're causing, where they're going to detach and get swept along with some of the intestinal contents down into the colon. All right? Once we get into the colon, we're going to start to get early encystment of the trophozoite. So we're going to start having a cyst wall being secreted. All right. Now, what's happening in the colon? What's happening in the colon?
Water resorption.
Water reabsorption, right? So for a long time, it was thought that the water, the dehydrating aspect, is what's triggering the encystment. But it's actually not. What's happening is we have cholesterol deprivation is what triggers it, which is associated with water reabsorption. Cholesterol deprivation is triggering the encystment of these guys. So we get the early encystment, and then when it completes, we have a young cyst, which has the completed cell wall. Now, this young cyst stage, we have two nuclei. All right, we have two nuclei. This young cyst will then develop a little bit further to produce a mature cyst. And a mature cyst has 4 nuclei. Four nuclei, 2 sucking discs, and 2 locomotor apparati. So we've kind of doubled, replicated, but it's not completely separated yet. The mature cyst then gets consumed by our host, right? And the cyst is going to excyst, or the trophozoite is going to excyst. I'm just going to say excisted stage, because at this stage, we still have our 4 nuclei, etc. Now, what triggers this excision? The low pH of the stomach. That's how it knows it made it into its host. So that's our trigger to exist. Once it exists, then, and only then, will we have the end of our division. So division happens in our cyst. We go from our young cyst that has 2 nuclei. We're dividing to get to the mature cyst, but we haven't finished dividing. We haven't finished dividing. That end process doesn't happen until we get out of that cyst, inside our host. And then we just continue replicating. Now, our hosts. I say this. Human. All right. And then the rest is the environment. All right. Now I can go even more and include some trophozoites that exit with the host. But I won't. All right. That's something you could probably do. Now, is the mature cyst the definitive stage? There's some argument about this. So it's thought that you could have a young cyst get consumed, that then finishes getting to the mature cyst and exists pretty rapidly after getting inside the host. That could happen, or it can happen, or you can ingest one of these mature cysts that then exists. So there's a couple spots, kind of disagreement on when. What the infective stage is. We do know that you're not going to get this unless you're at the mature cyst stage. And as far as what stage comes out of the host, that's the other aspect. Is it a young cyst that comes out, or is it a mature cyst? All right. In many of the pathology, you're getting trophozoites. You're getting different stages of cysts coming out. It's kind of presumed that if a young cyst comes out, it does have a chance of maturing to a mature cyst. Right, but again, we'll see. We'll talk about pathology. Any questions about this life cycle?
So we consume mature cysts?
We do, or possibly a young cyst. All right, so this is a monoxenous life cycle. Definitive hosts. The definitive host is human. Right, we said the low pH stimulates excision. So low pH of the stomach. Uptake of water in the colon stimulates these lost trophozoites because again, they don't really want to be there, but once they get there, they actually see it's cholesterol deprivation. Once they start getting deprived of cholesterol, then all of a sudden they start their encystment process. And the easiest way to identify young versus mature cysts is to identify how many nuclei they have. So in some of our cysts, on some of our slides, we can see 2 nuclei. Some of them, it's 4. On those 4 nuclei, in order to see them, you actually have to fine focus down through the slide, and what you would get is this cyst, and you'll have 2 that are there, and then 2 nuclei that are behind it. So you would have to fine focus as you move down through it. The back ones then become visible.
Does it have any intermediate hosts at all?
Nope, just humans. Well, humans and other reservoir hosts. But yeah, but no intermediate host. All right. Single stools of infected individuals can have up to 300 million cysts. That's what can survive out in the environment. Are cysts. In some cases, especially if you have this explosive diarrhea that's associated with this could have up to 14 billion parasites. That's cysts and trophozoites. As I said, these cysts are immediately infectious. They are. So when those young cysts come out, you could probably ingest one and get infected. Right? But in order to we still have to finish the division to get to our trophozoite. Get to our 2 trophozoites. And actually, I think you have to have several rounds of replication of these trophozoites before they can get to that stage where they can encyst. So there is some information about that. I'm not going to ask you. All right, questions? So how does this parasite evade the host's immune system? Quite simply, it outruns it. Very fast replication of these trophozoites. So much so that the cysts are being formed and are shedding from the host before an effective immune response takes hold. It happens quickly. Now, if left untreated, your body normally wipes it out in about 2 weeks. Normally wipes it out in about 2 weeks. But in some cases, you may actually get a chronic form. So this is going to be strain specific. So it's still Giardia duodenalis, but they have some different strains. Where one of them, it looks like your body clears it up, and then a week later, the diarrhea comes back, and you have it for a week, and then it clears up, and then you have it again. All right. This could last for months. Relapses could happen in as short of a week time period, or it could happen 2 to 3 weeks down the line. May be triggered by, let's say, stress of the host. Host gets stressed, all of a sudden trophozoites start replicating again. Giardia is most important for the immunocompromised individuals because their immune system isn't able to control the parasite and wipe out the parasite. So it kind of leads to dehydration, possible death from dehydration. Children are also pretty susceptible to this. So again, it's about the dehydration seems to be what the major issue is. But immune evasion, I mean, you do have some other things. Good. All right, so immune evasion was replicate as quickly as you possibly can. Host mounts an effective immune response, but trophozoites also exhibit antigenic variation. So it's somewhat similar to the trypanosomes. The process is different, but same idea is that you've changed your surface coat so that the humoral response no longer recognizes the trophozoite. Now, we have up to about 180 different antigens can be expressed over a 6 to 12 generation time period. And these types seem to be associated with both the infectivity, so how many cysts do you have to actually ingest to exhibit giardiasis, as well as the virulence, so how severe is your symptoms. So there's 2 things here, and it's like, what's the minimum parasite load that you have to get in order to exhibit giardiasis? All right, and also, based on that minimum, how severe is it going to be? And that's linked to your antigen type. All right, so I do have a diagram here. It's kind of hard to see. Maybe it might be a little bit clearer. And I apologize for the red and green. I have no control over the colors that appear in published papers. But they are targeting different antigen types. So you have a lot of antigen. The greens are hitting up some of the dominant antigen types, and you do get some blues on here. This is in early generation. Later generations, you start seeing some of these red antigens popping up. So our immune system is going to recognize whatever is dominant. It's going to mount the memory response against it, which is going to allow those subpopulations to start to increase in populations. That's probably where a lot of this relapse is coming from, the shift in the antigen types. All right. All right. So what we'll do is we'll pick up with pathology, Giardiasis, aka beaver fever. All right. Pick up that on Wednesday. We'll do our trichomonas, 2 species that we'll talk about in detail, a little bit detail, on Wednesday, and then we will move to our amoeba. And then hopefully on Friday is when we start our AP complexities. All right? Yep.
Excuse me, and the quiz is only on the parasites we went over last week.
Yes, the African trypanosome. So the intro to the protozoa and the African trypanosome.
And then for lab, sorry.
For lab, it'll go up through toxoplasma and plasmodium. We're going to save the Gregorins for next week because we're going to do dissection of worms. All right. See some of you on this afternoon.